Hey, hello everyone. Welcome to the mid-year Arise Town Hall. And in the Emil, could you mute, please? So I think this will maybe be one of the last Arise only town halls because Arise is growing, and it's also making a lot of friends, meaning adjacent programs that are stacking up, and we'll be discussing those too. So we have two special guests today as well. On the one hand, Rainier, who will explain about identity, and Niels Raas, who will explain about biodiversity meets data. If you have any questions, feel free to post them in the chat so that we can respond to them during the presentation. Just going back to the origin, and I know Hanko is also listening in, but many, many years ago, roughly 4 years ago, we actually started with building Arise, and the team looked quite different. We only had 5 teams. And by now, we actually have already 5 teams on the sample to DNA side only. And then we also have the sensor to digital species identification on the other side. So we're really growing. And we have a lot of expertise in-house throughout the organizations that we work with, which is amazing and highly necessary. And it also means that we are building towards that future. Arise is nearing its 5-year milestone, which means we go operational. And it also means that we're really stacking the programs towards this big integrated infrastructure that will help us to do species recognition, but also go into actual monitoring and creating data insights for policy, for industry, or for whoever wants to do something with biodiversity. But for Arise, and for most of the programs, it really starts with generating data, observation data of what species lives where. And that means that we really need that species reference database. Can we recognize a species? Do we have a specimen, an image, and DNA, sound, AI, and so forth? And that's exactly what we have been building. So without too much ado, let's move into the various teams that have been working on this. They will give you a short update of where they are, what they're planning to do, and what we have in stock for the next few months. Victor, can you take over?
Yes, the BioCloud. Well, first of all, I have to say we have a new product owner, Jessica, but she started a week ago, so it's a bit hard for her to present it immediately in the town hall. So I'm taking it over. Can you click when I point? Yes. So the BioCloud was also always shown as the brain of Arise, but I want to change that a bit. So if we we can better call it the memory of Arise in Q2. And the reason for that is, if we move on, we ingest metadata samples, we ingest lots of sensor data, and we also ingest DNA data. Basically, the BioCloud remembers that all. We have all that data coming from other brains, bringing us data, and we present that, click please, to the DNA dashboard and the DSA dashboard. What we want to change, what we are going to do now, upcoming, we've achieved this, we are there, and it's now time to put all this memory into effort. So what we're going to do in the upcoming months is we're going to bring back that data from the DNA dashboard into the BioCloud, where we can ingest the data that an expert adds based on the DNA dashboard. Think of new updated species information, identification, etc. In the DSI dashboard, they already do analysis using AI, and we are ingesting that data back into the BioCloud. Why? Because the BioCloud is a big memory dump where we have all that data, so we can present that to the outside world. But first, we need to get that data validated on international information sources. So we are currently working, as we speak, I think Luc is working on getting that data into GenBank and back, where we blast the information, which basically means we turn DNA sequences into validated species, families, taxonomy from resources in the world, the biggest banks out there. And if we have that information, we can finally do what we promised to do at Arise. We can bring that data to the world and publish it on both systems, DNA data, and on GBIF, sensor data. So this is the last part of the puzzle that we need to do for the infrastructure. And of course, there's quite some work to do to tweak and get get everything healthy and sane. But we're there, and we're almost there, and we will reach this around August, just before go real life. Exciting. So currently we have about 44 million sensor images with metadata, around 139,000 samples. It changes per day, so if somebody says it's a bit more, or then that's that reason. We processed already 80K sequences. We are currently still validating, testing everything together. If something goes wrong, we have to restart all procedures. But about 100 users, scientists are already bringing in data that we validate to find all these species. But a big change in the infrastructure is also, as Elaine said, we're not building this brain for ARISE, we're building a memory for biodiversity, identity, biodiversity, mid data, and many more programs will rely heavily on this infrastructure. I think that's it.
Let's move over to Emiel van Loon.
All right, I hope you can hear me properly now. And then let's move on to the next slide. I'll tell something about the monitoring and demonstration sites where the UVA team works on. Can we get the next slide, please? Where the UVA team works with partners at diverse locations to show how different sensors can work, how they should be deployed, develop scientific use cases. I will show something of that in a minute, and then also explore how these data can properly be connected to the BioCloud, be stored and used for various purposes. Can we have the next slide? So there's a huge array of sensors that we did connect, and in fact we try to continue some of these that are successful and we feel are important to discover more, and some of these are perhaps phased out a little bit because they're taken up by other projects, and we feel that we cannot add much more in our sites and projects, and we will develop a few new sensors or sensor combinations. I'll tell a little bit about the pilot projects we have to explore those. Can we go to the next slide? So first, an overview of all the sites that where sensors were placed. The black sites are the most important ones. Can we go to the next? These are the City of Amsterdam, the Oostvaardersplassen, the Waterleidingduinen, and the Veluwe. Can we move on 2 slides? And there is a few more. All right, and then, yeah, thank you. There's a few more sites that we are currently exploring. We didn't place any sensors there yet, but we have some people in the field who are able to help us. And the focus will, for the coming period at least, be more on acoustic monitoring in relation to bird migration with radar, and also in relation to field observations of various species groups. So that's the look into the future about these sites. Meanwhile, we continue monitoring as we did in some of the other main sites, one of which is the Amsterdamse Waterleidingduinen. I want to show a little bit about that because that's where over the past quarter a bunch of student projects have looked into innovation. So we looked previously at the interaction between, for example, rabbits and hares and the fox and the occurrence of the fallow deer. Can we move on? And now, with all of these cameras in the field, let's move on. And giving, a huge variety of pictures, of course, more than the few species we just saw. And we were especially interested in a few of the things that we didn't see so much on those pictures. Let's go to the next slide. One of the things that wasn't done yet and wasn't looked at yet was vegetation, in fact. So we have had a project that looked into the possibilities to use the same wildlife cameras for more diverse application. In what kind of habitat do we see? What can we quantify about the habitat, which is in this case mostly vegetation and other features of the landscape. And one of our students, Pien Kamp Meinert, she looked into the connection between these locations and the existing habitat map, as well as possibilities for using it, the information we have on these images for calibrating remote sensing, mapping of vegetation. In the future, we want to look at phenology and actual plant species recognition on our field cameras as well. So can we move on to a next? Because that shows a slightly different use of the wildlife cameras than what we did until now. We have tried to make camera traps that are more effective in observing squirrels. The red squirrel has been observed very infrequent and very little on our camera traps. So one of our students has investigated this specific setup suggested in the literature, which turned out to be not very effective in the waterleidingduinen until now, because we only observed these bird species visiting our site. This is, by the way, a baited trap, and not yet the squirrels that we aimed for. So we'll continue our search and develop a few of these kind of adjusted camera traps for squirrels, but also for rodents and small mammals in the future period. Can we go to the next slide? Finally, a third activity we had. Can we move on, or is it just my screen blocked?
We are at the rabbits, Emil.
Oh yeah, I don't see it. Oh, sorry. Yeah, so that's just mine. Okay, thank you very much. Yeah, oh yeah. I wanted, yeah, sorry, I'll move on now. So the final project that I wanted to show, but is apparently not here, is the interaction between fox and its prey animals, and it's the variation of that over the season. So animal interaction. Oh, there it is. Yeah, that was a project by Wouter Graaf, and showing that there are indeed opportunities to look at species interaction on our camera traps, and also relating that to activity data. So while looking at biodiversity and species that occur, it's also, we feel, important to use this information to discover more about ecological systems and interactions. And we've seen now that this camera network can be used in diverse ways for that. And in part, we try to move on and develop new sensors, and in part, we try to consolidate this and make this scale this up and bring this to a wider use, in fact, in the Netherlands. So can you move on to a next slide? Then the final thing I wanted. Oh yeah, let's move on, because I feel I'm running out of time, while continuing stuff that we have done, like comparing to transect monitoring and other existing observations, also the dropping counts, and compare this to our camera network. Can you move on for me?
I'm at the future plans, different camera setups, Emil.
Thank you. Yeah, so we'll try to combine these cameras with acoustic monitoring to learn more about small mammals, perhaps also looking into different ways to observe insects, because we still feel that there are challenges there, and that's going to be our main field-related work for the coming year, I would say, while also, of course, connecting the data that we collect in the field to the BioCloud, and doing useful things with identification and further processing. That was it. Thank you very much.
Thank you very much, Emil. And we're moving over then to the digital species identification. Laurens, can you take it away?
Yes, thank you. So I'm replacing Jacob because he's enjoying a sabbatical in Norway. But next slide, please. Yes, so in the DSI part, we start with the collection of the data, and we analyze the data using AI models, and we also make these results available to all users of the platform. And on the next slide, I will show you some of the updates we did regarding the launch. Please, next slide. So what did we accomplish since the last town hall? We had a workshop which has given us lots of useful feedback. We've also improved the synchronization of data from the BioCloud to the DSI platform. We have enabled the editing and creation of project sensors and deployments, and also you can now actually upload your own files to the system. We also worked a bit on accommodating international standards such as CAMSAT-TP, and it's also easier now to add AI algorithms. And we're working together with the UVA team to get the data from the MDS sensor portal also in the Arise platform, in the DSI platform, and the BioCloud. Next slide, please. So a bit numbers, you've seen a few already. So we have about 15 users at the moment. A few actually are recently active, so we need to improve that. We have 1,400 deployments, sensor deployments. We have about 44 million images stored. Most of them are Diopsis insect camera images. We support now 9 algorithms, 8 for images and 1 for sounds. And of those 44 million, we have analyzed 24 million Diopsis images, which yielded 92 million potential insects. So that's actually quite a lot of biodiversity, and we need to really dive deeper into this. Next slide, please. So a bit about the sensor portal integration is that we also now have the sensor portal as part of DSI, which means you can manage your project, sensor models, sensors, and deployments. And as said, also you can upload media items both through the front end, so there's the web interface, but also using a Python SDK. So for all those programmers out there, you're invited to also use this. It's not there yet, but before launch, we want to support a limited set of 4G-based sensors. Next slide, please. So one of the improvements we made regarding the upload is that if you now upload data which has weird stuff in it, it will give you a clear error message that tells you what to do with it. Next slide. And here you see a few examples of the interface for the sensor portal. Here you see a list of projects for a certain user. Next slide. You can inspect the project, right? There's also some, if you can see it like this, there's also some access control management, so you can actually tell what users of your, or what members of your projects can do what. Next slide. Again, you can also create deployments in the user interface itself now, which means you tell where your sensor is, when it and how long it will run, so that you have like this defined thing you can use for research. Next slide. And of course, I think it's nice to see this also all in a deployment map. So hopefully this map will at some point fill the whole Netherlands. That would be nice. Next slide. And as I said, we're working on this, and before launch, we want to improve a bit how you can add members and manage them. We also have some roles like an admin, an analyst, and an uploader. And this is all like, and we do some access control for this, so that you can actually have determine what member of the team can do what. Next slide. Also, we're planning to have a lot of user feedback sessions, right? So we also, if you're interested, if you're a researcher and you would like to try Arise, please send us an email, because we would like to have sessions with you to get feedback, and of course use this feedback to improve the platform. Next slide. Some other stuff we plan to do before we launch in the end of the year is to have a few new algorithms. For one, the Naturalis multi-source model will be there. This is the model behind Obsidentify and developed by Naturalis, and will be part of the Arise platform, so you can use it on your own data. We will add a few models from a research project called Mambo. And we also plan to have a big model from Google for wildlife camera images in there called SpeciesNet. And at the moment, we're also establishing the sync of the DSI information back to the BioCloud, so that we have a full circle thing. So it works two ways. Next slide. So something we would like to achieve for the DSI part. Before launch, we would like to onboard at least 10 test users, add 3 more algorithms, have at least 25 feedback items collected from you, and actually also fix those. And we are going to support at least one sensor type, the sniper camera or its successor, which I forgot what the name is, but please tell me if you know it. And next slide. And so after launch, right, so this will be in the 3 months after launch, we would like to have at least 25 more test users, add 5 more algorithms, and identify and improve 50 user feedback-based improvements. And we're also going to support more 4G sensor types, such as the actually not more sensor types. The Audimod doesn't have 4G, but the Bug has. And well, maybe we should discuss at some point what the third one should be. I think that's it from my side. Yeah.
Thanks a lot. And that concludes sort of the sensor to AI-based identification side of Arise. Let's move on to the sample to DNA barcode identification. Okay.
Yeah, thank you. I will present the results for the sampling team and also the apps team for Lotte van Leengoed. And actually, I'm very proud to be part of this team right now because it's a very big team. So we are with the team in the lab, but I also see us as the expert team. We have about 100 people helping us collecting the samples, and you are very much part of this team, and I'm proud of it because we are already having about 138,000 specimens handled in the laboratory. And this is something I will come back to later. Of course, how do we then still keep track of what we still need? Because we have so many specimens already in. Well, this is all BioCloud part, which we present in the other talks. But everything comes together in the online target list. I'm proud to share with you a common update on how the next target list will look like. In this, you can just search a species you find in the field and see where we still would like to have it. And then you can have a very nice way of filtering all the species that are in the Dutch species register to see whether we still want to collect it, yes or no. Of course, it was already presented before. We collect all the samples in the field, and we also need to collect the metadata, and this is what we do using our field kits that are brought to the experts so they can easily collect the samples and register everything in the ADA sampling tool. All the samples then are coming to the laboratory where we subsample them. So we take a part of the insect, put it in a tube, and we also make pictures, partly automated pictures, to store in the BioCloud and actually bring to you to the DNA dashboard. Here you see one of the fancy cameras, the Keyence camera we have for automatically photograph small specimens. And everything will come together in the DNA dashboard. Actually, it should work fine for all the users right now, but we know there are quite some issues, but please contact us when you miss samples, whenever you feel any problems are there, because we don't see them ourselves. We need your feedback to actually deal with the problems there. So in the dashboard, you will receive an automated update whenever there's new data coming in. This should work right now. So soon we expect new data, and you would receive an automated email for the new specimens that are having a barcode. When you're having your barcode, you can actually check it manually on online tools such as Bolt Systems, where you can paste your DNA barcode, and it will help you identifying or comparing it to a reference database. Be assured that this is not telling you which species you actually gave, but it's actually telling you to what references the barcode is actually looking like. But we really rely on the expert opinion. So when you say it's a certain species, when the expert says it's like species A, we still believe it, even though there may be a reference online telling us it's a different species. It may be a bad reference. So one very important part is that the DNA barcodes that we are actually generating need to be validated. And that's where our team, the expert team, is still needed. So getting back to the high number. So we have more than 138,000 specimens collected. More than 48,000 already of those specimens already have at least one barcode. And about 17,500 could actually be accurately identified to the species level, and it concerns 6,404 species. This was this morning. It means also we have a lot of specimens, more than 120,000, that were actually from bulk samples. These were not identified to species level, but they're partly barcoded already. And we need the expert team again to validate this, because when we have the barcodes, we will actually be able to say like which barcodes are new. We do not recognize them in any reference databases, and these new barcodes are actually probably specimens that we want to identify, and that's when we reach out back to the expert network. At this day, we need about still 14,000 most wanted species. First priority, we say established species in the Netherlands that don't have any barcodes yet. And it's actually in a very large variety of species groups, of which I highlight some of there. And the expert team we have, well, we have many species in the Hymenoptera, but only very few in the nematodes. So whenever you need experts that may help us filling this target list, please reach out to us. So how do we further fill the target list? We collect specimens from the field, but we also perform collection mining. So we use the collection from Naturalis we already have to actually see which specimens are there that we can sample, but some of them are very valuable or very old. We cannot use them all. So fresh specimens are still required. We identify the bulk specimens and we need to validate all the generated barcodes. So please reach out to us whenever you experience any problems. You are very much part of our team, so please, you need to know where to find us. We can use your advice, any recommendations, or anything else you want to share with us.
Okay, thanks, Onno. Kevin,
Yeah.
you're up.
Well, of course, between sampling and putting stuff in a dashboard, there's a whole workflow, which you'll hear more about now. So for numbers, we've passed the 80,000. Go to the next slide. We passed the 80,000 specimens processed in the lab now, and we're on track to go to 100,000 still this year. Lines going up steadily. But of course, these are all the new specimens that come in, but we're also sitting on a wealth of specimens, DNA extracts that we have already collected in the last 15 years, for which we had no prior success. So I had a student doing some work on redoing these DNA extracts, so redoing the PCRs and the sequencing from extracts that were sitting in the -80 freezer. We decided to start with plants, and for plants we sequenced 2 markers, one of which has some issues with primers, and one of which has some structural issues which make it impossible to sequence with the old Sanger sequencing technology. But now, of course, we have a new workflow and we found some new primers. So for both markers, we have more than 80% success rate on samples that failed previously. So even those samples that we already collected in the past can still be valuable and be added to the target list now. In addition to that, we have seen that many non-ARISE users are also adopting our workflow. So BGE, one of the European projects, also brought in some fresh specimens, about 15 plates that we've processed using the ARISE workflow. But also different researchers now in Naturalis. So there's about 11 researchers on various projects, from Caribbean beetles to European pollinators and algae, all using sort of the workflow we've now set up. So that's great to see, and we're hoping to expand that more in the coming year. Then also, of course, Westerdijk is working hard on the fungal biodiversity. So in the last couple of years, 500 soil samples were processed, delivering about 8,000 fungal cultures, for which they received more than 21,000 barcodes. And also at Westerdijk, scaling up is becoming a bit of a hobby. So as you'll see on the next slide, there's a lot of improvements made in the lab, which actually help them storing all these samples that they get from these cultures. Freezer space is always a bit of a limiting factor. Everyone that works in the lab knows all about this. But also scaling up the DNA isolations there as well. And so processing more and more samples each month. So that's great to see. And then, of course, as you all know, there's always nice pictures from new species being discovered. So if we look at the fungal species that have been sort of described within the ARISE project, there's about 14 scientific publications with 68 species new for the Netherlands and 50 species even new for science, which all came from about 200 cultures with 800 sequences. So you see there's a lot of work happening to also actually add new species. Nice thing is when we add these new species to the target list, we already have sequences, so we don't need to collect them anymore. So the target list is growing, but in a positive way. And here's some more interesting plates. Then for the outlook, so what needs to be done before we can actually go live? Well, I've already mentioned the legacy data, so that 15 years of history we have here at Naturalis at least. We're now uploading all of that to Bolt, so 30,000 records are already there. There's about 4,000 to go, and then we have to add some sequences here and there as well. And then we can finally publish all of that so that everyone can start using that data for their work as well. The laboratory information management system, the LIMS, is now nearing its final phase. So we're doing user acceptance tests this quarter. So we can finally sort of automate the registration process in the LIMS as well, and not just the DNA extraction processes, but also the registration of all of this, which takes a lot of time and has a lot of Excel sheets cobbled together, but that's all being replaced right now. And then the protocols that I mentioned before, we're also looking how to best publish them. We thought about doing a scientific publication, but that's a little more time than we anticipated. So we'll probably look at other avenues to get the protocols out there as quick as possible, because people are already asking for these protocols. Users of ARISE want to know what we did so that they can actually refer to it in their scientific publications when new species are discovered from the samples that they sent in. And then last but not least, identity. We'll hear more about it from Reinier in a bit, but you can see on this nice picture, we have more machines coming in. So all the equipment needed to process environmental samples are now installed. We have a system to transfer liquids in and out of tubes, which sounds silly, but it's a lot of work opening all these tubes by hand and pipetting, enough to drive you crazy. Some shakers and centrifuges to process these samples, and then also our sequencing machine. And we are now finalizing the protocols, which I will probably tell more about in an upcoming showcase. And that's
Thank you very much. Certainly not the last of the circle, but a very nice piece of the puzzle, Rutger.
Continuing on from Kevin, just want to make sure if I'm audible to the people at home.
Yes, you are.
Yes, you are. Okay, cheers. All right, so what does the bioinformatics actually do? First step is, well, on the left you see we have a bunch of cute critters, which we jointly are trying to sequence. These sequencing devices are now massively overpowered for the gamers, and that means that you can basically decode them in parallel. But because these are unknown things, well, how would you know what comes out in the soup on the outputs? Well, you know that because attached to the unknown DNA, there's known labels, so little bits of synthetic DNA, buy online. let's say. Those are attached to the unknown DNA so that once we are in the digital realm, everything has a little label so that we can then sort again what belongs with what sample. So this is just sorting. The expensive word that's used for that is demultiplexing. It's a pretty generic process, but it's the first step. So getting out of the soup what belongs to what sample. Next slide, please. Then in each of these outputs, there's very many so-called reads. So that's what's been detected by one of the sensors in the sequencing device. That's text data that has the actual DNA code and also some indication of the quality, because this is not a hit or miss kind of process. There's different quality levels in the observation. So the first step here is to trim and filter the low quality observations. Then what you also notice in the output is that within one specimen, there's a sort of substructure of sets of reads that resemble each other. Now, why is that? Well, our acute ladybug might have a parasite on it or something in its gut contents. So you might actually be observing multiple different species. Another reason for why you might observe that is that these barcode genes exist as functioning genes in a cellular organ called mitochondria. But those same genes also exist as kind of failed copies in the nuclear genome, so-called pseudogenes. Those are also sometimes caught. So there's a bunch of bycatch. And then the first step here is to do a clustering, which you see on the right. This is a pretty typical kind of picture that, for example, for one specimen, you might get 3 clusters. This part is being done by a pipeline, the so-called nanopore pipeline, which processes in batches. It's also available on our Galaxy portal for researchers, for students. It's been used in a bunch of events the last 6 months or so. And so it's kind of user-friendly. There's a graphical interface. There's also an automatic interface. So nanopore pipeline. Next slide, please. I just mentioned that there's these gene copies that no longer function as real genes. And real genes are under natural selection that make it so that it keeps functioning. If that selection is no longer there, then the gene just starts kind of mutating. And we can detect that, most cases, by checking if the sequence actually conforms to this model of the gene, because genes have, it's kind of like Morse code with start and stop, and you can check if that's actually present in the right location and if the translation to the protein product actually makes sense. So that's the first filtering step. We call that structural validation, which is again like a separate service, which also is on the Galaxy. We use it in Arise. We also use it in BGE, which was actually the origin of this tool. Next slide, please. So in addition to these pseudogenes, there's also this bycatch from potentially at least from other species. So now we want to know which one is actually the one that was on target and which is the bycatch. And so here there's a little bit of a puzzle. So imagine on the left-hand side, there's the true tree of life, which has species in it that might not even be known to science, which have species in it that have never been sequenced. So, which is why we're doing Arise, right? So we want to get a clearer picture of what's out there and fill the gaps in the face of an unknown true tree of life. So that's on the left. Then on the right, there's what we are collecting within Arise. And so sometimes we have a species like here, our cute little ladybug, which has never been sequenced. So how would we know if that's the right sequence? Well, what we can do is look at the family that it belongs to. So these barcode genes are inherited by evolutionary descent, so closely related species will have similar sequences. So we have sort of a picture of the expected kind of genetic diversity within that higher group. And then the check that we can do is see if what we have now observed fits in that picture. So here I just want to also second Onno's point. This whole technology can't like positively say it is this. It can just say, well, we're in the right direction or not. Of course, it is still interesting to know what exact identification came out. And if it's a species that has been sequenced before, then we can actually get an exact match, but that's definitely not always the case. So treat these kind of results with skepticism. Here, the context is just we want to see if we're on target or if we're just looking at gut contents or something like that. So again, here, this is like separate tooling, and this tooling is also, we can go to the next slide, that's fine. So this is again separate tooling, which is also available as a web service. So here, bringing it together with the BioCloud. So what we're building with Team BioCloud is that this service can be invoked in batches, so that when everything has come together with the sampling information, where the experts have said, well, I think it's this, with all the lab data that's needed, metadata that's needed to actually do the sequence processing, when that's all come together, then we can do this validation. And so what we're now trying to get into like actual production is that BioCloud can invoke this. The principle works, but we're ironing out the last details, including what to actually present in the dashboards, so that it's clear to end users what they're looking at. So that's the number one thing on the outlook here. Then next, as Kevin already said, we want to bring everything, of course, to Bold, get it out in the real world. So that's a work in process. There's a bunch of things that need to happen for that. One of them is that within ARISE, we anchor our species on the Dutch species register. Annoying thing about taxonomy is that it has existed for at least 200 years, and that taxonomy is just formalized opinion. Taxonomists have lots of opinions, and the different databases sort of represent these different opinions, such that, for example, if we submit to Bold, they might have a different species name, like a synonym for the same thing. Also, the higher organization might be different. So what we're working on now is to address those potential mismatches so that Bold understands what we mean when we come knocking with our data. Next thing is in this whole process, we have to address some data quality issues. Of course, when you bring it all together, you notice that, well, a bunch of this metadata along the chain has been captured in spreadsheets by humans. To err is human. So sometimes there's some issues there where once you start checking, actually errors bubble to the surface. So we're addressing those issues, rerunning the batches so that it's all ready when we go live. And then also so that we're ready to actually start submitting. And so the final of those three items there is that we're together with Team BioCloud building a standard workflow that basically checks all the boxes for a complete, rich upload to Bold. And then lastly, what's kind of coming up, Kevin alluded to, is this laboratory information management system, which is used by the people in the lab to enter what they're doing. And that's much more structured than spreadsheets. So, of course, we all expect that some classes of quality issues will go away. On the bioinformatics side, that means that we're going to start talking to this system automatically to then harvest a whole bunch of important metadata, such as the actual code of these known little labels that I mentioned earlier, the synthetic DNA, so that people don't have to copy and paste up within spreadsheets. It's just in a database. And so we get rid of those type of just copying errors, stuff like that.
Thanks.
Thank you very much.
Thanks, Rutger. Super cool. And you can hear by the energy in Rutger's voice that we're really closing the circle now. And it also means that we are really preparing for a official going live. Now, obviously, ARISE already has many components that are operational. People are using it. We're interacting with all of you, with hundreds of people. But we really also want to make this kind of official because end of October, we have had 5 years to build ARISE and we really want to celebrate. We'll give you the details and what it's going to look like, but it will also involve this sort of front end platform page, the home of ARISE, where you can enter and log in or find more information and really interact with all of those components of ARISE that we have been building, much more kind of glued together in that big puzzle. What I promised was also an outlook on what's next of ARISE. So I'd like to give a view on the first two that are mentioned here, identity and biodiversity meets data. So Renier, if you're ready and you have a mic nearby. and please, in a nutshell, explain us what identity is all about and what you have in store for the upcoming months and how it connects to Arise.
Yeah, so identity. With identity, we have the ambition to become, as Naturhaus, to become a biodiversity observatory, to reveal changes of species and ecosystems by detecting eDNA or by using eDNA. We aim to deliver a national standard for biodiversity monitoring using standardized methods and scalable measurements like meta barcoding to detect the invisible in soil, water, and in air. So we started with soil already. With the data, we want to support, we will deliver high quality data using, of course, the Arise reference database, and thereby support policy making, scientists, etc. Where are we now? Currently, we work hard on the sample management part. So the front side will be very important to handle the thousands, 10,000s of samples we need to analyze. As you heard from Kevin, the lab facility is ready or becomes fully usable next week, so we can start with the first experiments. There has been done a pilot for air eDNA, which is very important because there's a lot to be. There's a lot unknown yet for air eDNA, so we need to define there also a standard for identity. The sampling strategy is almost finished. Very important to discuss with stakeholders to understand how many plots we can sample and how much work there is for the stakeholders. In the next 6 weeks, we will onboard a new stakeholder. That's most likely to happen in Dunea. We also are at an early stage of onboarding Natuurmonumenten and maybe even Rijkswaterstaat. So in the coming weeks, because we have the lab facility ready, we will decide on the soil DNA isolation protocol, conduct additional experiments, start first sequence runs of the samples that we have already in from Bosgroepen, and of the Leiden Biodiversity Network. And then following will be then. And also Bosgroepen is really enthusiastic, so they even asked to help us more, and more stakeholders are asking to help and add to the success of identity.
Thanks a lot, Reinier. If you have any questions, feel free to reach out to Reinier van der Geest directly or any of us, and we will point you the way to identity. Niels, are you ready and online to explain a bit more about biodiversity meets data?
Yes, I am. I hear an echo. Is that okay? Good. So biodiversity meets data. So we kicked off this project in March this year, and biodiversity meets data will scale up on all the developments of Arise. So we're gonna bring this to the European level. What biodiversity meets data will do is to build a single access point to high throughput biodiversity monitoring in combination with AI species identification algorithms, mobilizing biodiversity data and developing biodiversity analysis tools for better conservation across Europe. Next slide. So there's a number of expected outputs. So this is a very short pitch, but so the output is here, a better monitoring of biodiversity in the European Union by high throughput methods and the usage of existing data. So we're going to mobilize a lot of historical baseline data to GBIF and bring it into the analysis pipelines in order to deliver a better implementation of the European nature directives, which is the Bird Directive and the Habitats Directive. This should lead to a better understanding of the state of nature and the drivers of biodiversity loss. So we're going to investigate how to mitigate these negative impacts on biodiversity, and that should result in a more complete view of the state of nature and its evolution in Europe. And next slide. So, and then here is basically what the single access point will look like. So that's in the center there. Then on the top left, we have the spatial data catalog, which will contain a lot of spatial data on abiotic and biotic conditions, which determine the distribution of species and represent the drivers of change. Then the lower left is the plug-and-play deployment of high throughput monitoring devices, and that's where we scale up on all the developments that the RISE has brought, using camera traps, audio devices, and eDNA sampling. In the middle bottom, we have mobilization of baseline data to GBIF. So the baseline data go to GBIF, but also the high throughput biodiversity monitoring data go to GBIF. Then the top right, we have a WebGIS viewer to consult all this data and to consult the results of the VREs. And the VREs are represented in the lower right. So we're building analysis pipelines to analyze terrestrial, freshwater, and marine biodiversity from local to European scales. And that's basically what BMD represents in a nutshell. Thank you.
Thanks a lot, Niels. Excellent pitch. And if you want to know more, feel free to reach out. Again, it can be through us, but also to Niels Raas directly. Well, let's close off this presentation. It's a review of Arise and where we are, but of course, the big goal for all of these programs and this extended team is to work towards knowing exactly what the state of our biodiversity is. Only when we start measuring those individual species beyond the habitat directives, but have a full-fledged view on biodiversity, do we actually know what's happening and what we can do about it.
I have a question for identity in the chat.
I'm going to close the recording now. But we're still open for questions. And again, feel free to reach out.